Welcome. I'd like to welcome Jason Bobe. He's an associate professor and the director of the Sharing Lab at the
Yep.
ICON Institute for Genomics and Multiscale Biology at Mount Sinai Hospital. He leads the Resilience Project and is founder of two science nonprofits. He's a very busy guy, personalgenomes.org and DIY Bio. And today, Jason's going to talk to us about sort of a sorted mix of your projects, right?
Yeah.
Yeah, and so he's also a Mozilla Science Fellow and a really cool guy. So I'll let you take it away, Jason.
Well, thank you very much. I'm delighted to be here. So can you see my deck?
It's a little small.
Okay, so you know what I'm going to do? I'm going to go and change the screen sharing setting to my whole desktop here. Let's see, screen share, entire screen, share. How's that?
That looks great.
Awesome. Is that better?
Yeah, that's great.
Okay, cool. We'll have to do this again towards the end because I actually have a second shorter deck that I want to go through. But I really wanted to start with some of the ideas that I'm passionate about because it's really framed a lot of the projects that I've worked on. And if you're interested in open science, I think you'll find some of those things interesting as well. And let me say that you know, at any time, if you'd like just to stop me and ask questions, or if something's not clear, just feel free to interrupt me. So my name's Jason, and I work on a whole lot of different projects that I love very much, and I'm going to talk about each one of these individually a little bit. But one of the things that really started my interest in biomedical science was really a fascination with DNA sequencing technologies and the belief that in some ways they were the X-ray vision goggles of our era, because they, you know, it has this magical ability to take invisible things and make them visible. And so there's been tremendous change in how people look at genome sequencing over the past 15 years, 20 years. And so, and this is driven by some pretty remarkable innovation in the actual technology itself that's driven down the price by several orders of magnitude. It's really just incredible. Very similar curve, even faster than Moore's Law, which I'm sure you know. And in the early days of human genome sequencing, this is the kind of infrastructure that you needed. And it was an activity that was really only available to experts with advanced degrees in world-class institutions like this one is the Whitehead Institute, who was involved in sequencing the first human genome sequence. And then over the years, these devices themselves are actually becoming, much smaller, more affordable. They can now fit on your desktop, and an early version of this one even had an iPhone dock. And some of them are getting so small that they are, effectively portable and disposable, like the Mini Ion sequencer that you plug it right into your computer and the data streams right into your computer, and you can drop your solution into the machine there. And this is nanopore sequencing. I'm not going to talk a whole lot about the technology, but I'm really interested also in some of the innovations in technology that are coming from the other direction and from a grassroots way, because I think it paints a picture for us about how the world's going to operate over the next 10 to 20 years. And so this is a picture of a young man's hobby laboratory in his basement. His name's Fred Turner, and he's the 2013 UK Young Engineer of the Year. And he basically bought the components and built a PCR machine in his basement. And so this is really a kind of innovation that the DIY bio movement, it's like my friend Matt Cow calls it, innovation at the trailing edge, and that in many ways, there are pieces of technology out there like polymerase chain reaction that's been around for decades, that now there's a whole established set of makers and hardware hackers who are turning their attention to these, really interesting laboratory devices that are sort of old and stable in some way, and are refashioning them and making them interesting again. And so, and this is really happening across a whole set of tool chains, where it's becoming more of really thinking of biology as something that you could actually do as a hobby. And there have been Kickstarters where people are, backing, some pretty advanced laboratory devices with the goal of making a sort of consumer version, low-cost technologies. And there are a variety now of venues where people who, I would say, access points to technology in ways that really expand the numbers of people who are able to engage. I mean, this is a group in Cambridge University called Hack Reduce that was, basically providing training, classes, and access to high performance computing to people in the Boston area. And you could go there without necessarily any skill and learn to use Hadoop, or and use some of the tools that were otherwise only available to experts. And then the same is actually happening with in biology and GenSpace. This was started by a few friends of mine in 2009, I believe. And they actually just got their first major foundation grant last month from the Arnold not the Arnold Foundation. It was a major foundation here in New York. And this is, you can kind of think of, the gym that you belong to, and maybe you pay $100 a month to go to the gym and to belong to a gym and to use their exercise equipment, where they kind of have a similar model where you can pay a membership fee and get access to their infrastructure for doing biology. And then they would also offer seminars, classes, courses, workshops, and those kinds of things. And GenSpace is one of a number now of community laboratory spaces out there who are offering more education and access to people to learn about some of these exciting technologies, which are really changing sort of all aspects of our lives, from medicine to agriculture to material science. And GenSpace, another story, they also had a collaboration with a group, an academic group, where typically this was I believe it was the group who was doing the looking at the subway microbiome. And the punchline there was basically, one of the things that they found was, I don't know how many of you have ridden the subway, but when you're standing on the train platform and the train's approaching, and there's this big blast of air in your face, as the train's pushing, a column of air, that's basically just a big cloud of rat dander. There was a rat DNA on every surface of the New York City subway. Well, anyways, they went around swabbing all these different surfaces. And so, so think about that the next time you're on the subway. But they went around swabbing all these surfaces, and they needed to actually, extract and purify and amplify the DNA to do the analysis. And they contracted out to GenSpace to do some of that work, because they were actually, people who were just interested in doing PCR, right? That, they could actually go out to the community, have members of the community extract and amplify this DNA, which was just a sort of standard practice. And rather than paying, an industrial lab or to do it for them, that they would actually, it was like part of the community engagement strategy, that members of the community would help actually do parts of the science. I think that's really interesting. And so let's see here. Whoops. Hang on one second. There it is. And this is just one of the projects that I work on is connecting an expert group of safety and biosafety experts who traditionally would service, academic and industrial labs, and making their expertise available to people working in the community or at community lab settings or in, hobby workshops, so they can get access to safety advice. But so now that's just sort of setting the stage, I think, for understanding what the next 10 or 20 years of science looks like, because in many ways, I think science is as Stephen Friend, a colleague of mine, likes to say, it's this guild of experts. And there are a lot of forces that are breaking that down and making science and people who are trying to reclaim science as something that's really accessible and available to everyone. And obviously, open science, the concept of open science is really, part of that. But there also are stories that, to me, really indicate how exciting it is, and the opportunities that are ahead for making some of the connecting these technologies and expertise to broader audiences. And this one story about two high school students. Well, first of all, let me say, this story also connects back to sort of DNA sequencing being part of, having X-ray glasses, because you might say to yourself, you go to the grocery store and there's really no mystery there, right? You don't need X-ray glasses. All of the foods they're labeled. They tell you what it is, and it's got you can look at it, and it's got the prices right there, and you just would ask for a pound of whatever fish that you want. Well, these two high school students went around New York City, and I want to say this was like 2008. It was the year before GenSpace opened, because I remembered they did this work at Rockefeller University, and like, if it would have been the same two girls, several years later, they probably would have done this work at GenSpace in Brooklyn. But that wasn't available yet. That concept of a community lab wasn't even really invented yet. And so they did their work at Rockefeller University, and they went around to grocery stores and restaurants, collecting pieces of fish and writing down what it was being sold as. So got some yellowfin tuna over here, and got some tilapia from this store over here, and so on and so forth. And then they took it back to the lab, and they actually did DNA barcoding to determine, to compare what the DNA said what the type of fish was versus what it was being sold as. And what they found was a lot of misrepresentation of the seafood that you would buy in stores and restaurants. I mean, and when I say a lot of misrepresentation, I mean like really gross levels of misrepresentation. I can't remember the exact statistics, but it was like, in somewhere at least around the 40% of fish being mislabeled. And then and things like, really hilarious things like sturgeon caviar, which was like $1 gazillion an ounce, and it was actually Mississippi paddlefish eggs. And people were really being straight up dishonest, or somebody had been bamboozled along somewhere in the food supply chain. And so then some investigative journalists in the city of Boston heard about this story, and then they went and did their own version, all driven by the work of these two high school students with access to these technologies. And they went around Boston, and same story. And this actually led to a change in state policy to address this issue. And this was then, and again, this goes back to citizen science, and I think that's really exciting. So moving on to more serious topics, the mislabeling of fish, there are also early examples of people who are sick and tired of waiting passively for other people to solve their own health crisis, and where maybe they've tried the conventional medical route and just have been unable to to be able to get connected to the right experts, or for people to take interest in their problems. And so they've gone out on their own and have set out to try to solve their own health problems. And there are actually a growing many instances increasingly like this, and some really amazing ones. And this is one that started I don't know, a number of years ago. And Hugh Reinhoff is an interesting guy. He isn't your average citizen scientist by any stretch. He was actually trained by a really famous geneticist, and so he really knows what he's doing. He has formal training, but his daughter was born with a variety of problems that concerned him. And no doctor could say, well, it's like the reason why she has curved fingers and she has some unusual traits, they could never pin it down. And he was worried that maybe under the surface, maybe there was some heart issue or liver issue that if he didn't know what it was and couldn't address it, that it would actually reduce her lifespan of a really happy and lovely girl who I've met personally. And so he wanted, as a dad, to do everything he could to help understand what it was, so that he could create an environment where she could thrive and get the most out of her life. And so one of the things that he did, after running into many dead ends, was to actually set up a lab in his basement where he could more affordably basically extract her blood and purify and clean the DNA and amplify it, and then send it off to clinical labs to do work. And so he was partly and he convinced members of the larger biomedical research community to join him on this mission to identify what the causal factor was in his daughter's syndrome. And he since has made and published his findings on this. So another transformation that is something that I pay a lot of attention to, and really where I focus a lot of my work, is on this concept of participatory research. And I'm going to come back to this concept when I talk about the Resilience Project, but maybe I'll just start by saying that I had the good fortune of working with this scientist in Boston named George Church. And he wrote this editorial in Molecular Systems Biology in 2005, where he sets out this sort of ambitious goal of recruiting a cohort of individuals who would be willing to forego their privacy and have basically all aspects of their biology decoded and made a public resource, so that experts and citizen scientists could have access to this really, really rich data set that would then be useful for going and developing, computational models and visualizations, and getting people who could make major contributions in advancing biomedical science, but otherwise, wouldn't typically be trained or have access to this kind of data. And those would be, like mathematicians who are working outside of a U.S. biomedical research institute, or computer scientists who have no idea, aren't trained necessarily in human subjects research, but might have really great contributions they can make if they could get access to ethically published public data, and so on and so forth. And so I spent quite a few years helping to prototype and develop the Personal Genome Project with George, and then a few others. It's now in 4 countries. So. And one of the things that's really interesting, and so here are some of the, so now we have 4. This is just, there are 4 countries. The Personal Genome Project did a lot of things that were novel all at once, and one of them that's really important to me is this concept of equal access, where if you're not just going to be a human subject in biomedical research, but you're going to be a co-investigator. And for that to happen, you actually need access to your own data. And so people who signed up for the Personal Genome Project got to see the same data that the scientists got to see about them, their own data slice. And then also there's a lot of other things, but I want to draw a comparison really to this is a comparison actually that was first drawn by Misha Angrist at Duke. And I just actually noticed in this last picture right there in the front row is Ellen Jorgensen from GenSpace in this group photo from an event we held a few years ago. But so, you know, in the old-fashioned model of biomedical research, the old-fashioned model was a sort of fight club, you know, where the identities of the members are hidden. And so the experience of trying to operate a research study where the scientists aren't able to, you know, directly interact with the research participants in rich ways, or the research participants are not able to interact with other members of the research study, it really makes it very difficult to actually build a community that's interactive and human. And so, you know, there are now, I think, many researchers are starting to understand that there are other benefits that, you know, by participating in research, people, you know, one of the things that people might actually look for is community as a value, of a reason why that they would participate in research. And so I think that's pretty exciting. And so equal access, I already talked about this. So this is the idea that you get a copy of your data in the research study. And another project that I work on that I was co-founded with Madeline Ball and the software engineer Bo Gunnarsson, that they both have just done an incredible job over the last year building out this platform. And so something cool happens once we sort of are able to break the exclusivity of having a research scientist own your data and that research scientist be the sole person in charge of making decisions about who's able to access it, right? And you know, who's qualified to access it and what other initiatives matter. So when you actually get a copy of your data, you can actually make those choices yourself. And if there's some other research study that you find is valuable, or even if it's not a research study, maybe it's Wikipedia, maybe you want to contribute your data to some other effort that completely, you're empowered to do that when studies practice equal access. And so an example, a prototype of sort of what that future looks like when equal access is a practice that is widely adopted by medical research communities. You know, it's possible then for individuals to basically create longitudinal integrated research profiles that can combine a research set from a variety of modalities, whether or not it's your health record and your participation in the Personal Genome Project and a brain scan from some other study. And rather than each of those being data silos that only a couple people can control, there can actually be long-term sharing of integrated data sets through things like this. And this work was supported by the Robert Johnson Foundation. And that foundation, I encourage you to go check it out. Manuel and Bo are doing really incredible work. And this is, so this is sort of the end of my first presentation. And so I'm going to take a breath and just switch over to talking a little bit about the Resilience Project.
Okay, do we have a question?
Yeah, sure, ask him.
Go for it.
Yeah. Oh, I'm here. My first question is, does the lab that you described, do they ask any, you know, to be an author in the paper, or if you do your research in that lab?
Oh, like the
Gene.
the gene.
Oh, GenSpace?
GenSpace, yes.
Oh, you mean does GenSpace ask for co-authorship?
Yes.
Oh, that's hilarious. I doubt it. I am like 99.99% certain that the answer is no. And it's also true that I think I would also say that people who sign up and participate at GenSpace, they're not necessarily driven by the same values as an academic publish or perish research scientist, right? They may they may be just curious, or a hobbyist who's interested in learning how a piece of technology works, right? Or they actually, in some cases, and in many cases, they're professional scientists who, they wanted sort of in their spare time to work out some idea that they weren't able to do in their industrial job or in their academic job. And so on an evenings and weekends basis, then they have the freedom to do that. And I think also, so I would just say that the answer is probably no. I highly, highly doubt it.
Do they provide chemicals as well?
Yes, I think that they, these places often have, they provide basic infrastructure, and then also the infrastructure to, they don't pay for your reagents. I think often you would be responsible for paying and getting the reagents shipped to the lab. And then, each of these labs also, they have they have infrastructure and types of experiments that they would support, sort of BSL-1 versus BSL-2, that kind of thing. So they have processes for reviewing your project and making sure that it's something that is appropriate for the setting.
Okay.
Anyone else?
Okay, so I just wanted to spend a couple of minutes talking about the Resilience Project, and I'm spending a really huge amount of my time helping to incubate and produce the next phase of the study. So we just had a publication. Well, actually, I'm going to get into it. So I'll get into the retrospective and the a little bit later. But first, can you say if anybody is actually enrolled in an organized health research study? Can you raise your hand or actually make some kind of noise? Because I can't see you right now.
We've got a big 0 here.
A big 0, crickets. Hey, guess what? Guess what? This is actually the same pretty much everywhere I go. So, and it's embarrassing fact of today that, and I largely speak to biomedical research, professional biomedical research and medical communities, go to conferences where there are people who are doing some kind of research in health and biomedicine. And I ask the same question, and usually there's like 1 or 2 people would raise their hand and say, oh yeah, like I'm signed up for a study. But for the most part, almost always less than 10% or even less than 5% of people are engaged in biomedical research. And to me, it's a really interesting paradox where if you ask people what are things in the world that matter to you, and this was actually something that was done by the UN for their sustainable development goals, they actually went out and surveyed people in something like 120 countries and asked. And here you see health is a number 3 priority, and that. And it's also true that it's on the Maslow's hierarchy. It's something that people know is important. And they also, when you look at professions that are trusted, healthcare workers, whether it's a clinician or a nurse, they are highly trusted, among the most trusted professionals out there. Scientists have a little bit more work to do. They're kind of in the middle of the pile. Lawyers and advertisers at the bottom. As you probably also suspect. But people trust doctors, people care about health, but nobody participates in health research. At this point in history, is completely a fringe activity. And even in cases where, where there is the most traction right now in participation in biomedical research, those are like very severe, fatal, chronic diseases. But even in those instances, participation is still incredibly low. Like, for example, there was just some news last week that claimed that only 3 to 5% of eligible people fighting cancer sign up for clinical trials. And so our ability to advance medicine, which supports health, which we all care about, is really driven by participation in research, which nobody is doing. And so this is a major problem. And one of the things that is really exciting about the Resilience Project is it has an opportunity to be an invitation, possibly the first invitation that people will get to sign up for biomedical research. Because I think people's mental models around biomedical research is really the cancer clinical trial. And they're like, oh, well, like that's not for me. Like I'm, I don't need to sign up for a clinical trial. It's not for me. But really, human research is something which is just much broader than that. And there are a lot of really important types of just understanding basic human biology and basic research about human biology that people could really contribute to and advance our ability to enhance our wellness. And so the Resilience Project has a message to people, which I think is really intriguing, and it asks them to consider the situation where, your biology could actually hold the key to curing somebody else's disease. And the hypothesis that we have is that we've been missing a whole stream of insights because those who do participate in biomedical research, they tend to get in the front door by going to a doctor because they have a health problem, getting diagnosed with the disease, and then learning that one option for them is to contribute to a research about that disease. But everybody else, they never get that invitation. And so we're missing a stream of people, of healthy people, who actually could help us develop new pathways to prevention strategies and even new therapies. And this is really turning the sort of disease research on paradigm on its head. But we're searching for something that's really kind of special, and we expect it to be rare. And because it's not just a healthy person. In some ways, like nobody's completely healthy, but we're looking for a fraction of the healthy people, the healthy people who, in fact, medical textbooks say should be sick, but somehow they've escaped and they're healthy. But they may have no way of knowing that they should be sick because they're healthy. They don't have the typical signs and symptoms of the disease. And at this point in history, they probably also don't have their genome sequence, which would indicate to them that they carry this risk factor or these risk factors. And so this was an example. This is an example of somebody who's really intriguing, and that was profiled in the New York Times recently. And this is an individual whose family is really devastated by an early onset form of Alzheimer's. And I forget exactly the number of his relatives that died early and before, in their 50s from this early onset form of Alzheimer's, which is genetically driven autosomal dominant disease. And because his mother had a big family and his mother died, and I think many of her siblings died, and he's watched this disease really ravage his family. And then but he kept getting older and without having signs and symptoms. And so, to everybody's surprise, including his, when he did get tested, he actually carries the same mutation that in everybody else that we've seen, it causes them to get sick. And so the question really is, why is he special? And can we go out and systematically search for people who have some kind of protection against disease? And this is another example. This guy's name is Stephen Crone, who unfortunately died a few years ago, but his story lives on, and it's really an incredible story. So his partner was I think the first person in the United States to die of AIDS. And this is when we didn't really understand the disease. It got sick in the late 70s. And so then, as we figured out what HIV and AIDS, what they are, he then realized, oh my gosh, this is an infectious disease. I'm going to die the same way. And so went and, went to the Jared Diamond AIDS Center and got tested for HIV, assuming that one day he would get positive and then succumb to AIDS. And those test results kept being returned as negative. And this caused was a great surprise to him, he has this quote here, "I don't know why I was surviving," which I found in a documentary about him from the 90s. And he was legitimately, he sort of suspected that he was resilient, which is, I think, fascinating. And so did the clinicians that he worked with. And so somebody smart said, "Hey, can I have some of your blood? I think we should actually look what's happening. Look at your cells in a dish." And they took some of his cells and they gave it a massive dose of HIV in a dish, and those cells were not infected. And they're like, "Wait a second, we made some kind of error. We need some more blood." And so they repeated the experiment. Sure enough, no HIV infection in a dish. So like, okay, something's going on here. And as they went and were looking at Stephen Crone's biology, they made a discovery that he had a protective genetic variant called CCR5, or this molecule is called CCR5, and it lives on the cell surface. And so then they started trying to figure out what this molecule, like, how does it work? And basically it's a docking station for HIV that lives on the cell surface. And the HIV requires this docking station to then integrate into the cell and do its damage. And he had a version of it that just like wasn't working. So HIV couldn't dock, and was, his cells in some way were like Teflon coated, they just bounce right off. And so then a couple of drug companies jumped into the mix. And Pfizer in 2004 actually got an FDA approved drug that basically tries, to reproduce what this effect of what Steve Crone had naturally, you could now get in a pill and share that with other people. And I think this is a really inspiring story, because it's another example where it's possible that we could actually go out there and create more stories like this, that we could find more people who were resilient to disease, and then find new avenues to therapies. And we don't actually know how common resilience is going to be, particularly if you really want to think about it at scale of looking at all diseases, and all people, if it's going to be something that's super rare that you see, 1 in a million, or if it's going to be something that, you actually might find examples of it, 1 in 10,000, who knows? And so we're going to go figure that out. And there are 3 steps. I'm not going to, go into all of the steps for this call in great detail, other than to say, basically, we first have to go out and do a search. And once we find somebody that is a resilient candidate, we would then want to validate that they do in fact have a genetic mutation that increases their risk for a disease in a meaningful way, and then that they lack the typical signs and symptoms, phenotypes of that disease. And once we then have a validated candidate, we would go on to do the decoding and look at really sort of the whole level of all from their genome and their RNA expression and microbiome, if it makes sense, medical records, and try to then do the work of finding the needle in the haystack. What I like to call it, a smoking airbag, as opposed to a smoking gun, because it's like, what's the thing that's giving them the protection that's buffering this disease? And that becomes a really interesting and challenging scientific problem to do that. And so, really, we're basically what the part that I've been working on is the bat sign. And the prospective study, we're going to allow people who think they are resilient to a disease to go sign up and tell us about it. And then we'll put you through a process that we can then, validate or invalidate your case from a scientific perspective, and then decode you if it makes sense. And then also, for people who have no idea whether or not they're resilient, it's still possible that we could genetically find examples of where you carry risk factors that you may not know about, that and that you lack typical progression that in ways that are interesting and valuable to the rest of society. And so there are 3 ways that we search. The thing that we got a lot of press this past week because we published our preliminary retrospective results, where we looked at the genomes of around 600,000 people around the world from a lot of partners. And one of the things, this is a shout out to Christy. One of the problems actually of doing a retrospective analysis like this is using other people's data. Often they don't have everything that you want, and so you just kind of have to live with it. And do a lot of work to get the data in a format that you can actually analyze and make discoveries. And often it just is missing some percentage of the things, the data points that you care about, but you live with that and you move on. And so that's what we did. And in the end, of the 600,000 people, there were an initial set of around 15,000 resilient candidates that in some way, the algorithms said, hey, you want to look at these 15,000 people. And the person who actually went and manually looked at all 15,000 cases and then put them into bins of levels of support and or just saying not valid because it was a resequencing mutations because it was a sequencing error, or noting that we didn't have access to enough health information, or for whatever reason, we whittled it down to this list of 13 individuals who we could really confidently say these are really strong, resilient candidates. Now let's go try to talk to them and see who they are and hear about their stories. And one of the other problems with having an unhappy ending was that we were unable to basically contact any of these 13 people for a variety of reasons that are that really are unnecessary. And that's really sad. And so that's another reason why going out and prospectively recruiting individuals to sign up, I think is going to be an important effort. So I'm not going to really talk about the decoding. I mentioned it. We did invent this concept of a decoding board, and this was something that was really Stephen Friend's idea, I think. But basically, it's modeled after a tumor board, and because like in cancer, it's really complicated, right? So you might have a patient who has had a tumor biopsied and they've had some blood work, and then you might want, the treating physician might invite an oncologist and a pathologist and some other experts to join in reviewing the set of data to try to come up with a treatment plan. And so our decoding boards are doing something very similar, but it's asking a different set of questions, which is basically not why is this person sick and what do we do about it, but it's why is this person not sick, and then what do we learn from that. And so that's really exciting. We held our first decoding board meeting here in New York City this past summer. So basically, I'm spending a lot of my time on the design, and we're just transitioned to the build, and soon later this year we'll launch. And I'm not really going to talk much about all that went into that because it's complicated. I mean, in my 30-second pitch is that to do this well, you really need 4 things. Minimally, you need 4 things. You need a lot of things, but you need, of course, good science, right? And you need good governance, and you need to utilize the right tools and platforms. And then you also need an engagement strategy that speaks to people. And doing any one of those things well is really hard, and doing all 4 of those things well is nearly impossible, and is not something that the biomedical research community really does well at all yet. So there are a lot of challenges in there. But anyways, we really want to make this not about resilience in childhood disease. That was really our pilot study, but we want to make it about resilience in all diseases. And we're going to have an app, which I won't really talk much about, but just to say that I really feel like excited and grateful to be in a position to bring biomedical research to new audiences. And basically the whole world is a new audience because nobody's participating. But if people do, we actually really do have a chance, I think, to change the world. So lots of people to thank. These are a few of them. In particular, 2 of my mentors and the head scientist on this, Eric Schott and Stephen Friend, and then many others. So maybe with that, I'll just pause and thank Christy for inviting me, and take any questions that you might have.
Thanks, Jason. That was awesome.
Yeah.
You go sign up for biomedical research now?
I guess I'm not an active participant, but my gut microbiome is part of the human gut project.
Fantastic.
Part of the what project?
The human gut. Is that what it's called? Human gut?
American Gut.
American gut. Yeah.
Oh, awesome. So that reminds me. So I'm running an event at Harvard Medical School April 25th that brings together 18 participatory research studies. And American Gut's one of them. Rob Knight is an old friend of mine who is the PI of that study, and they'll be there.
My sister-in-law was working as a technician in his lab at the time.
Oh, cool. What's her name?
It's Emily Taravist.
Emily Taravist, great.
She's a bioinformaticist.
Awesome.
Yeah, but he's moved to San Diego, I guess now, right?
Yeah. Oh, that was at Colorado?
Yeah.
Yeah.
I don't know how to interpret it. I got the data, but
Give it to me.
I don't think no one knows how to interpret it.
Well, Madeline, actually, we're running a workshop in San Diego on May something, in a couple of weeks, as part of the Health Data Exploration Project and the Quantified Self Public Health meeting. And then the third day is a data dive. And in one of the workshops that we're having is how do you actually take raw microbiome data and use the tools to make meaning out of it. It's not easy.
But you've got a little project now.
Yeah.
I have a question. So for the biomedical survey or research, is there any requirement or preference on the biodiversity of the ratio, the races and the locations of the people? Because I've heard that there are a lot of genetic diseases, they have a certain geographic patterns of some diseases are only in maybe Southeast Asia, some only in North America, some in Africa. Is that a consideration in this kind of study?
Yes, it's certainly a consideration in interpreting. So this is a really big deal in understanding the impact of genetic variants, where often in the United States, the information that we have is from, Caucasians only, or mostly Caucasians. And so you can't necessarily apply this information to then everybody else in the world, right? I mean, we know this from the goals of precision medicine are that, people have a unique combination of traits, and we have to tailor our health towards that. And in science, it really also creates problems around interpretation if we have, large excluded sets of types of people, right? So diversity, I think, and understanding and having really rich knowledge about how human biology works, that's really important. I mean, in terms of requirements, I mean, I think that it is a goal of every research study to recruit as diverse as possible, and that there is, and so people are really interested in making research studies more attractive to people who are conventionally, do not participate in research.
It's kind of surprising that this is, to me, this is surprising that people weren't doing this kind of thing sooner. Is it looking at the, this other treatment group, I guess, people who didn't get the disease? Does it have more to do with just having the data now in order to do this?
Yeah, I mean, I think that, well, I think if you think about it for a second and think about the ways that people typically get into biomedical research, it kind of makes sense, right? So that why we haven't studied people more broadly. So there's a couple I think reasons to talk about. One is if the way that you get into a biomedical research study is that you get sick and you go to your doctor, and if your doctor happens to be part of an academic medical center maybe, or a large teaching hospital, then not necessarily like your community hospital's probably do a lot less research, right? But if you are a person who, you know, goes to a larger medical center that does do research, and you go there because you're sick, that's how you actually get the invitation to join research. People aren't out there actively looking. It's not a cultural activity yet, right? Like people talk about, hey, what'd you do this weekend? You know, it's not like, oh, I signed up for a health research study, right? Right. But I think it could actually get there. I think it could be something that we do. But we have to actually think about what the value paradigms are going to be for people. And it can't just be because I'm an altruist, because I have so much free time that I just want to, this is what I just want to do, right? I think if we start to expand the set of things that people get back from health research is that they get their data, that they learn something, right? And they get educated about issues that they care about. They get real metrics that their contributions are having an impact. There's, you know, lots of things that can be done. But one of the reasons why it hasn't been done yet of this study is because the on-ramp to biomedical research right now is that you are sick and you go to the doctor because you're sick. and that's how you get in. And if you're one of these individuals who are resilient, that may never happen to you. I mean, a doctor and a healthy person walked into a doctor's office, they're like, great, you're healthy, get out of here, go live your life, right? So, and then I think the other reason is, I think, you know, the NIH and a lot of funding is rightfully so targeted at sick people, right? Targeted at people with disease. If you want to study cancer, then you need some tumor samples to look at, right? But there's also, that's not to say now that that there are real opportunities to also incorporate, now that we have way better tools at way lower cost with things like smartphones, that it's way more conceivable that we can actually go out and reach millions of healthy people versus the old-fashioned way of biomedical research only happens in hospitals. You have to go talk to a doctor and sign a paper consent form, and you know, all of that. Like that's not going to scale.
I guess I just would have imagined that health researchers would have, you know, be sitting around thinking for a long time, like, I would love to get my hands on some healthy people data.
Well, that's something that we do in entomology too. We go to fields where there are insect problems.
Yeah.
Like that tends to be the focus of our work. We never really take a look, and we're really, really bad at counting insects when they're not there, or, you know, writing it down when they're not there. You know, we have go to a field and say, okay, well, let's check on the infestation problem. Oh, we don't see anything. Okay, well, we don't need to record it. And so, yeah, when we're trying to look at sort of the broader ecology of a pest or any insect species, we really have very few absences in our data. Yeah,
too.
Well, yeah, that's true. Yeah, so it's kind of a common problem in biology, you know, especially in the applied biology. Well, any other questions for Jason?
Are you anticipating a lot of paternity suits?
No, yeah.
Or something.
Not actually. I mean, the data that I've seen on that is that some of the expected numbers, like there was some inflation that I forget exactly sort of who started the rumor that paternity rates are, you know, 15% or something, like really like, oh my gosh, that high, you know. Of course, it's a reality. It is a reality in the world, but I think there is data to show that it's not as common as people worried about initially. I mean, in the very early stages of humans getting access to their own DNA, people, or just medical information generally speaking, people genuinely were worried in all kinds of ways. That, you know, that thinking that humans, generally speaking, were far too delicate to be able to shoulder the burden of learning anything about their own biology, right? And if you go back and look at the debates around HIV testing and home HIV testing, people were then claiming that if somebody learned their HIV, they would jump off a bridge, right? And I think a lot of that was actually based in, obviously real concern for people, but a lot of those were not actually did not actually reflect reality. And the same is true for and then other people take a sort of moralistic tone about learning about your own biology. And so an amazing article in Consumer Reports in 1979, maybe, gosh, I can't remember, I have a copy of it, which was home pregnancy testing. There was a quote, and this was the first Consumer Reports review of a home pregnancy test, and the editor basically said something to the effect of, it was like a quote from a public health official, like, any woman who doesn't want to go to the local public health department to get tested obviously has something to hide, It's like, what? And so there's a whole lot of that going on too, generally speaking, around access to data. In some cases, I think genuine concerns for people misunderstanding things. In other cases, people are taking unnecessary moralistic tones. In some cases that bother me, but
Wow, I kind of want to go back into the past and rough up that public health official.
How do you feel now? Yeah.
He's still alive. Nursing home.
Undoubtedly it was a guy, right?
Oh.
Like undoubtedly it was a guy. A crusty old guy.
Yeah.
It's like totally not hip to the times.
Oh, well, the tides have changed. Well, that puts us at 4 o'clock. So thank you so much,
Thanks.
Jason. This
Oh,
is,
you're welcome.
and good luck with all your planning with the conference.
Yeah, thanks so much. And good luck with the course. Thanks again for having me, Christy. And good luck to you all.
Bye.
Bye.